So we look at physiology of the eye and vision. Kindly take time to check out the anatomy video on the eye. It might be very helpful before you start looking at this. So first of all, the principle of optics. We should know how the light rays travel. So normally they are refracted when passing from one medium to another due to differences in density. And that might end up refracting or bending the light. So typically parallel light rays striking a biconvex lens are normally converged. Okay, so they are refracted to a focal point on the retina. So, and this is how a biconvex lens looks like. So it will actually converge this light and it will actually be focused at the retina. So the point of focus is normally called the focal point, and the distance between the lens and the focal point is what we call the focal length. So ideally, it's not only the lens that bends or refracts light, as we said, just moving from one medium to another bends the light. But now for the eye, light rays are bent all over, starting from the air to the cornea. The cornea bends the light, and you can see it has a refractive index of about 1.38. The fluid, or in the aqueous humor, the aqueous humor bends the light also. The lens bends the light, the fluid in the vitreous humor, until now the light gets to the retina. So there are a bunch of things that help in the refraction of light. So it is not only the lens. So another concept we need to know for physiology of vision is accommodation, and this is a very important concept. Basically, this is an active process that occurs as a result of muscular actions that affect the size of the lens or the curvature of the lens. So normally at rest, objects that are closer than 6 meters, or what we call 20 feet to the eye, will appear blur. So this blurriness is sorted out through what we call accommodation. So what is this accommodation? So it is the process by which the curvature of the lens changes in order to basically focus light at the retina. And as you can see here, a far object will require the lens to change its curvature. A near point object will require also the lens to change its curvature, so that the ultimate event is that the image is actually formed at the retina. So if we look keenly at now the accommodation, so in this case, you can see the lens, and it is held by the ciliary ligaments, or what we call a zonulus. So when we are looking at, when the ciliary muscles are relaxed, so these muscles are relaxed, they will pull the zonulus, therefore pulling the lens and making the curvature become a bit thin. So this is accommodation that is necessary for distant vision. The other concept is if we are talking about the ciliary muscle contracting, so the zonulus becomes less tense, then the lens becomes thicker, or the curvature becomes thicker. This is a good kind of curvature for near vision, so it will be able to bend the light even more. So the other thing is basically the elastic lens is attached to the circular ciliary muscles, as we know, and the muscles are linked to the lens by the zonulus, which now is made up of elastic fibers. These are the fibers that are able to either pull or relax, so that we change the curvature of the lens. Now, pupillary reflexes. So the pupillary reflexes, mostly we look at two. So we have pupillary light reflex. So this is basically a simple concept where when light is shown directly onto the eye, that specific eye that is getting the light will actually constrict, okay, to reduce the amount of rays that is coming in. So in bright light, the pupil will become smaller and smaller. It will constrict, what we call meiosis. But in dim light, the pupil will become bigger, and the purpose of that is try to get as much light inside the eye as possible. So that would be a dilatation of the pupil. We call that mydriasis. We also have another very important concept called consensual light reflex. So it is when light is shown on one eye, the pupil, the effect on that pupil should be seen on the other pairing eye. So if you put a torch if you shine a light from a torch on your right eye and the pupil constricts, we also expect the same to be happening on the left eye, even if the left eye has not received the same light. So they work together. That is what we call consensual light reflex. The other important concept in physiology of vision is a field of vision. So the field of vision basically is that area that is seen by the eye at any given point. So basically, field of vision is how far you're able to see from basically centrally and even peripherally. Okay, so the area that you're able to see without you having to turn your head. So that is what we are talking about, a field of vision. So if you're looking in front or ahead, so every other part that you can see, so either centrally or peripherally, that is what we call a field of vision. So typically a normal eye should be able to see approximately 95 degrees on the temporal view, what we call towards your ears. So going to the side, okay? That is 95 degrees. So that is what we call the temporal field of vision. And approximately 60 degrees nasally. So 60 degrees nasally is basically towards your nose. So that is going inside, okay, from the center. So this is what you're talking about, 60 degrees nasally and 95 degrees temporally. So, however, at the point where we have the optic nerve, that area, normally at the optic cap, we don't have photoreceptors. Therefore, at that point, even if light falls on that area, there is no image that will be formed. So typically that is normally called a physiological blind spot. However, we normally even don't notice it because the eye is able to now take the sensory receptors, the sensation from the previous images that it was able to form in the environment, and then it makes you visualize even that area to the point that you might not even notice actually that you're having a blind spot. Then we have binocular vision. This is a type of vision in which basically we deal with animals that have two eyes, and we know human beings have two eyes. And when you're facing the same direction, the two eyes typically form each eye will form its image. However, they come to a point of convergence, and that is now what we call a binocular vision. So the field of view, which is the area that you can see when you close one eye, will overlap significantly from one eye to another. So the center of the eye's field of view overlap with one another, and this portion of the visual field provides basically the most detailed information because both eyes are able to view the same field. So that convergence provides the most information, and that will be the most clear sight of vision as compared to the areas that are only viewed by one eye. So for example, the right eye will have a view of the periphery, but the left eye will not be able to reach there. So, and the same goes for the left eye view. But the center bit where both eyes are able to view, or they overlap, that is the perfect area for vision. So the range of focus here, we don't have plenty to talk about, but we know, and we've talked about accommodation, 20 feet or the 6 meters is the distance that we are able to see without requiring any accommodation. Okay, but if we have an image less than 20 feet or even more than that, then we will require some sort of accommodation for us to have a very clear image. So then we move on to a very important concept in vision, what we call the visual pathway. So what exactly happens from the time that light bounces off your eyeball. So the visual pathway is anatomical structures that are responsible for the conversion of light into electrical action potentials. As we know, when light enters the eye, and then it is refracted and focused on the retina. From that point on, the light has to be converted into electrical impulses that are able to be carried by the optic nerve up to the visual cortex in the brain. Okay, so that whole route is what we call the visual pathway. So as I said, it begins in the retina and ends at the primary visual cortex. So basically, it begins at the point of the retina here, and then carried all through to the visual cortex. So the pathway is as follows, as you can see. So from the retina, it will follow the optic nerves. So the optic nerves will cross each other at the optic chiasm. So from the optic chiasm, you will have optic tracts. So the left one and the right one. So they will end up passing through what we call the lateral geniculate bodies, and then we form optic radiation. So the optic radiation will end up at the visual cortex area, and that is where we actually now perceive the image. Okay, so if we go step by step, what happens in each area? So we know the retina. If you again take time to look at the anatomy video, you get more content on the retina and the structure. But the retina basically consists of receptors that are able to convert light energy to nerve impulses. So the retina normally has two quadrants, if you have to say that. So if this is your retina, so you have the part that is closest to the nose, what we call the nasal retina. So this side and this side, because the nose is around this area. And then you have the parts that are closer to the side of the head. So that is called the temporal retina. So this is important because, for example, the nasal retina of the left eye will be able to see the left visual field, okay? Because that is where the light will be coming from. And the nasal retina for the right eye will again see the right visual field, as you can see. So but the right eye's temporal retina will be seeing the left visual field, and that goes for the other side. So it's important because this helps to know how the light from different parts of the retina are carried forward. So retina, as you said, has several layers. So we have the retinal pigmented epithelium. So this is just the first layer which absorbs light and prevent light from bouncing all over the retina. So it absorbs the light, and also it's responsible for providing nourishment and supporting basically the neural retina. So the neural retina is made up of three other layers: the photoreceptor cells. So these are the cells that are involved in converting lights, photons into electrical impulses, basically what we call phototransduction. So this is the point where you will find the cones, the rods. Okay. Then you have bipolar cells, which now these ones relay the converted electrical impulses from the photoreceptors to the ganglion. So this is like a tube of nervous cells that just relays the electrical impulse from the photoreceptors to the retinal ganglion. So the retinal ganglion, these ones now form part of the optic nerve. So they receive the electrical impulse from the bipolar cells, and then together they form part of the optic nerve. So they are the part of their heads form part of the optic nerve. So this is the pigmented epithelium, okay, absorb the light. Then you have the photoreceptors. So you have the rods, you have the cones. So these ones are the ones that convert now light into electrical impulses. The impulses are carried through the bipolar neurons, and then the bipolar carries them to the ganglion cells, which form part of the optic nerve. Okay. So the optic nerve, so we have gotten the impulses from the retina and all the layers. So the optic nerve we basically have so many optic nerves. So we approximately have 1 million myelinated axons that arise from the ganglion of the retina, and they come together at the optic disc. So at the optic disc, you have now the optic nerve that passes through. As you can see, this is a bunch of optic nerves that are coming from the eye. So we know that at the point where we have the optic disc or the optic nerves are coming in, that point we don't have photoreceptors, so we have a physiological blind spot. And the tubular nerve exits the orbit. So this is where they exit the orbit of the eye, so through the superior orbital fissure. So once they leave the eye through the optic, so we have the optic nerves. So they come and they actually converge at a point we call an optic chiasm. So this is not only a point of union or convergence, but also a point of decussation. So basically, where we have that is where they separate each other. Okay. So for example, if you look at the nasal retina from the left eye, it will come. Sorry. So if you look at the nasal, the nasal retina, so the impulses are being carried from that point, and then they end up on the right side. Okay. And then the nasal retina impulses that are carried by the optic nerve from the right side are carried, and they end up in the left side. So as you can see, the nasal ones are able to cross the other side. However, the temporal, the impulses from the temporal side, they don't cross. Okay, these ones don't cross, so they remain on the side that they were supposed to be. So the nasal fibers of each eye, they cross the midline, joining the temporal fibers of the contralateral eye, or basically of the other eye. So following the decussation, so the visual input from the right half of the visual field will travel to the left optic. So we're saying that because if this is your right side, the visual field where the nasal retina is looking at is half of this right field view. But you see, all of it will be carried to the left side. The same thing, if this is your left eye and you have your nasal retina here, and it's looking at this half of the field view, the rest of it will be carried to the right side. Okay, so that is the point you need to know. So while the stimuli from the left half of the visual field will pass through the right optic tract. Okay, so once the optic nerve passes the optic chiasm, it's no longer called the optic nerve. Now it's called optic tracts. They are now called optic tracts. So optic tracts basically are the compact caudolateral continuation of the retinal ganglion. So they are carried through some tracts here, and each tract carries fibers that are stimulated from the contralateral visual field, basically. So this is now what carries the impulses now from the chiasm up to now, where we have what we call the lateral geniculate body. So this is an important, this geniculate body is very important. We have two of them. They are round shaped and located laterally and they have protuberance. Basically, they are like protruding. So they house a multi-layered lateral geniculate nuclei. And this is very critical, very important for processing of vision. So the processing of vision starts from here and using the nuclei that are found on the lateral geniculate. So once the vision has started being formed from the lateral geniculate, now we have fibers. So these are very many fibers, and they call optic radiation. So the optic radiation, they represent the visual tracts that extend from the lateral geniculate body, so to the primary visual cortex. So the optic radiation is separated. So we have the left side that ends up in the left visual cortex, and the other one that ends up in the right visual cortex. So this one is separated into two main tracts that correspond to the upper and lower quadrants of the visual field. So finally, Once the radiations get to the visual cortex, so the visual cortex basically is normally found at the back of your brain, basically located at the occipital lobe. So the visual cortex is the area of the cerebral cortex that processes visual information. So the area of the visual cortex that receives the sensory input from the lateral geniculate bodies or nucleus specifically is called the primary visual cortex. So yes, we have the visual cortex, but the specific area that receives information from the geniculate nucleus is primary visual area. That is the point that we now actually form images completely. So the visual cortex has two areas. So we have the left hemisphere, which receives signals from the right, and the visual cortex in the right hemisphere, which receives signals from the left visual field. And this happens, as you know, because of the chiasm that we had. So ultimately, what we all these things that we have just talked about, the entire visual pathway, is that when you have an image or an object that you want to see, it will be refracted and then formed. The light is focused at the retina. But at the retina, because the light is inverted, we will have an inverted image. Okay, but remember, when you look at an object, they don't look upside down. And that is the reason is because the light now is converted into electrical impulses, passes through the optic nerve, okay, gets to the optic chiasm, then it's able to dissecate. Then we have the optic tracts, okay. The optic tracts again carry the information up to the lateral geniculate body, and then we have the optic radiation, and then we end up forming the image at the visual cortex.